Thank you very much, Filippo. Thank you, Professor Gao, for organizing this important session. And as Filippo emphasized, and as I will show you in part of my presentation, our interactions with Chinese science is very strong and is growing stronger every year. Within cardiovascular research as a journal, we focus on clinically relevant discovery science. So we want to publish studies that provide novel discoveries, but with clinical context of clinical samples. And I will show you examples of studies within this that we recently published that well exemplify this breadth of our interest and breadth of our focus. Over the years, our impact and influence over cardiology field has grown very significantly. When we started about 6-7 years ago, the impact factor, for example, was about 4.5. We managed to increase to 13.3. And while this measure, of course, is not the most important measure of the journal influence, I think the interest that the journal receives from the broad group of areas within cardiovascular sciences and really a growing potential for submissions in very, very broad group of topics is evident for the journal. For the papers, what do we expect from high-impact papers in discovery science? We expect, of course, impact, we expect novelty, but the robust methodology, translational potential, reproducibility, and most importantly, mechanistic insight are essential components that make a perfect cardiovascular research paper, as some of the examples that I will show you exemplify. As I already mentioned, and as Filippo mentioned in his introduction, the importance of Chinese science for our journal is enormous. As you can see, 48% of total submissions in 2025 so far are originating from Chinese scientists. So thank you very much for these contributions. It is also important that not only that the submissions are rising, but also the acceptance of Chinese manuscripts is rising by 25% over the most recent 3 years. And we have very tough acceptance rate because at the moment our journal is receiving 2,500 papers per year, which is a very big strain on some of the journal, and especially after the recent increase of impact factor, we had a flood of papers, which caused some slight delay in the month of August, but now we are back to quick handling of the manuscript. We have associate editor and editorial board members from China. You can find them on the website. And, you know, I think that we really emphasize the high visibility for articles from China. People Republic of China is one of the top article contributing and citing countries in the journal within the last 3 years. And you can see this is the rise of submissions from China between 2019 and 2024. Is very spectacular and very strong, and I want to thank all the Chinese scientists for this contribution. Our positioning within the cardiovascular journals of discovery science is growing, as you can see, but what I always emphasize in this point is that our growth is associated with increase of number of papers that we publish. So in fact, you know, while many journals on this list have decreased their number of papers because they want to maintain high impact factor, we have taken an opposite route, and we have increased number of papers because I believe that market share in business is very important feature of your business. If you have high market share, you are important. If you have low market share, you're not important. And I think in journals, number of papers we publish, high quality, of course, is a representation of market share. And that's why I'm very proud that, you know, even if we have some variations of impact factor in next years, it is going to fuel our growth in the future. It is interesting that not only China is the top contributor, but is actually also at the moment becoming top user of our journal. 24% of downloads of articles in cardiovascular research are originating from China, mainly as everywhere around the world, our reviews and original articles are being downloaded. And now some examples of top research. And one of the top contributing research papers to the impact factor of 2024 was actually originating from China. As you can see here, heart-gut microbiota communication determines severity of cardiac injury after myocardial ischemia and reperfusion. And these researchers have combined, as I mentioned, important point always, the combination of human study with the discovery science. So in STEMI patients, they have identified that increased levels of gut bacterial translocation products are associated with left ventricular function and with other indices of myocardial viability in clinical assessment. And they looked at the clinical predictors of this in a well-powered clinical cohort. Then they moved from this question to a number of different models. I will only show you some figures because, of course, as with every paper now, we have many figures and a lot of studies. But the most interesting ones are the increase in intestinal permeability upon the myocardial ischemia reperfusion injury that leads to gut microbiota translocation. And you can see the presence of signatures of gut microbiota in the heart, in the spleen, in the blood, and all over. And this can be detected on both the level of LPS and the level of the bacteria themselves. Then they show always an interventional. This is another key aspect of a good paper, is that intervention is used to reverse the observation that is being done, and the pre-depletion of gut microbiota ameliorates ischemia reperfusion injury. We can see this here, and they compare the SPF mice, which are living in good hygienic condition, I would say, with completely germ-free mice that are pre-depleted with gut microbiota. You can see that ischemia reperfusion is lower in the depletion of gut microbiota, and this is associated with lower inflammation. It is associated with number of systemic changes, including fibrosis as well. But while this was contributing, it was a previous paper in 2023 that can be calculated to contribute to the impact factor last year. We have number of papers from China that are very, very highly downloaded, and I will present examples for you here. And as you can see, our journal is not shying away from good quality clinical trials as well. And this, you know very well that recatizumab has been just approved, I think, in China in May, and this coincided with the publication of this trial in our journal originating from China, showing the effect of this anti-PCSK9 novel antibody that originates from China, that we published the clinical evidence in phase 3 study 4. And then some discovery science. The transcription factor Y1 regulates vascular resistance and blood pressure dynamics through epigenetic control of the M6A RNA modifications in vascular smooth muscle cells. The authors have generated a number of different vascular smooth muscle specific knockout mice to track the mechanism of action of YY1 on blood pressure, and these are traces of blood pressure measurement, and you can see that the YY1 knockout in smooth muscle cell is associated with diminishment of systolic and diastolic blood pressure, especially at the beginning of induction of blood pressure increase. And the same is occurring when you knock out one of the downstream genes, MYKK. 2. So in smooth muscle cell as well, the authors track the whole mechanism quite well in a multiplicity of figures, and I want to congratulate them for this highly downloaded paper. And then, of course, we want to look beyond just blood vessels. We publish papers on different components of cardiovascular system, including platelets. And in this very interesting paper, team of Professor Jung Boguet have published that the Phetuin A increases thrombosis risk in non-alcoholic fatty liver disease, and explain the mechanism linking it to TLR4 expression on platelets. The effect of Phetuin A picked up and initiating signaling coming from TLR4 receptor within the platelet instigates and initiates the activation of the cell. And this seems to be a mechanism quite specific for non-alcoholic fatty liver disease, the metabolic liver disease, that provides a link between the liver and cardiovascular system on another level than typically analyzed. And then further looking in cardiometabolism, we have some unexpected observations that, for example, tirzepatide, the Mounjaro, actually can be very effective in experimental models in alleviating the cardiotoxicity exerted by some of the chemotherapy drugs, in this case doxorubicin-induced cardiotoxicity. And the mechanism, again, the team of Ji-Yu Ling has provided a very clear mechanism for this through HRD1-mediated NRF2 ubiquitination. So these examples from studies from China are showing how highly downloaded and highly impactful papers look like in our journal. Every year we also award something called John Schilling Award, and we award this globally to the way that this award is awarded is top 5 impactful and downloaded and cited papers are scored by reviewers, and the top scoring papers receives the award. The award is given at the session together with European Heart Journal during the European Society of Cardiology meeting, and a plaque is given, and also some money is given, which I am very proud, because most awards nowadays don't come with money, and ours is coming with some degree of money. We can't give too much, but we try to support the researcher and their team with some funding as well. And this year, Jolanda van der Velden presented a study of her lab led by Paul Winker on hypertrophic cardiomyopathy dysfunction mimicked in human-engineered heart tissue and improved by sodium glucose cotransporter 2 inhibitors. And this is very interesting because they show that SGLT2 inhibitor actually could provide a therapeutic window and therapeutic potential for hypertrophic cardiomyopathy. And as you know, in hypertrophic cardiomyopathy, we really don't have too many options at the moment. And you can see that they have generated a model based on iPS cells that mimics the biology of human myocardium. Most common genetic heart disease, hypertrophic cardiomyopathy, frequently cause of sudden cardiac death, increase in left ventricular wall thickness, impaired cardiac relaxation are key elements of pathology that is observed in this disease. But many people, while looking at mutations that are responsible for the disease, are forgetting that the picture in these mutations are actually leading to a number of changes in physiology and pathophysiology of cardiac myocyte. And these changes include some ionic changes with increases of intracellular sodium and increases in diastolic calcium. And this is very important because SGLT2 inhibitor is known to potentially modulate those. And that was the hypothesis of this important paper. The inhibition of sodium hydrogen exchanger, NHE, will lead to decreases in intracellular and sodium, decreases in intracellular calcium, so will be potentially beneficial for the hypertrophic cardiomyopathy changes within the myocardium. And the authors asked the question, is SGLT2 inhibition beneficial in early hypertrophic cardiomyopathy, and if so, what is the mechanism? And indeed, they have shown in the mutated patients in iPS cells and iPS cell engineered tissue, based on these cells, that there is improvement of features of relaxation and of empagliflozin, and the degree of effect is shown here. We can see a very clear distinction and effect of empagliflozin. They looked at very many mechanisms, and this was like a detective work. They look at SGLT2 inhibition, no effect. Inhibition of NHNF 1.5, no effect. Lowering cytosolic oxidative stress, no effect. Via phosphorylation of contractile machinery or calcium channel, no effect. And then in the end, the NCX channel, the NCX inhibitor, SEA, seems to be reverting these effects, suggesting potential mechanism for this observed effect of SGLT2 inhibitors, which is, I think, very important observation that will lead to future potential more targeted drugs to be used in hypertrophic cardiomyopathy. And it is indeed led to subsequent study that was published in European Heart Journal from the same team showing the effects of empagliflozin in animal models. So in our journal, the iPS cell human heart tissue was published as an initial one, and then in vivo evidence followed up in European Heart Journal earlier this year. Then the last paper I want to show you is the top-downloaded paper published just in June 2025, coming from the group of Isabel Goncalves. And I want to show this to show you that our journal not only publishes the mouse studies, but we also focus on clinically relevant discovery science. And this, in this study, the authors used the Swedish cardiopulmonary bioimage studies, CAPIs, to investigate large number of coronary CT data in the context of dietary information and diet that patients have used. They have identified and divided patients into patients with high dietary index and low dietary index. You can see high dietary index is good, a lot of vegetable, and too much hamburgers, low dietary index. And they have shown, first interesting thing is that clearly women eat better than men, because in the Swedish study, the distribution of dietary index in women is much better than in men. So it's indeed a good thing, I suppose. And they look at number of different factors. I had the pleasure of writing an editorial for this, which we called "Let Food Be Thy Medicine," which is a very famous quote on when we related it to anti-inflammatory diet, where they identified that intermediary effects on triglycerides, waist circumference, and hypertension are occurred with high dietary fiber intake and low dietary fiber intake, causing an opposite effect that results in key features of coronary plaque in this very large, nearly 20,000 CT scans that were analyzed. And then they also provided validation in subsequent studies in a Scott Hart study from Scotland, adding to the strength of this evidence. And very last thing I want to draw your attention to, and I want to invite you, because at the moment we have an open call for cardiovascular research early career reviewer program. So we want to bring on board young investigators in order to pair them with our editorial board members, with our seasoned reviewers, to prepare them for working with our journal on a routine basis. Each participant will complete a 2-year program with minimum number of reviews to complete per year. And then from this program, we are going to develop this into junior associate editor program. But we decide not to start immediately with associate editor program, but slowly bring people on board with the journal. The closing date is very soon. So please, if you are interested, submit or send to your colleagues and invite, because we would really love to have at least one of our early career editor reviewers from China itself. So I hope I have shown you the way that we try to shape the impact of the journal. The word cloud of the titles of our papers is shown here, showing that we really cover the whole breadth of cardiovascular sciences. And with this, I will end and thank you for attention and for opportunity to present here today.